itself for this nice right. So, first I'd like to thank Professor Atalia for giving me this invitation and giving me this opportunity to share our work. So, today I will be going to share my recent research which fits this title, that is Supramolecular functional activities with temporal and spatial. So, basically, I work in the area of supramolecular chemistry, that is physical chemistry. So here I will talk about how the assemblies can be controlled in a time and in space between. Space is what I am saying, not that space, it's just within a particular location. Well, so I think as it is in the department of physics, we may not cover what is fundamental assembly. So I like to give a brief about it. So the Kolkata chemistry was introduced by Professor Raychaudhuri, and academically it is the chemistry beyond molecule. So the chemistry of molecular assemblies using non-covalent bond and interactions, that is the supramolecular chemistry. Now what is supramolecular assembly? That is the spontaneous organization of molecules in order structures, non-covalent interactions. What would be the non-covalent interactions? This could be a hydrogen bond, electrostatic interaction, bistatic, anaerobic interaction, and others. So if we compare the strength of the bonds with covalent bonds are much stronger than these non-covalent interactions. But when these individual non-covalent interactions they work together, the force is sufficient to hold the structure in a stable position. These are some common examples of supramolecular acid. So this is the alien double strand structure. This is basically hold together by base pairs. Micelle, you may have heard of the term micelle. Basically, lipid bilayer. Enzyme substrate complex. And another interesting one is that is mitochondrial. I will talk a bit about this one later. So, well, the type of this collaborative way is actually come into these three categories. One is static acid, that is, the structure once it's formed remains stable for longer time, not change over time. So it actually lacks the time scale property. It simply points to the energy minimum. What happens in this case, the static acid. So your monomer or building blocks, they are significantly higher than. And the atomic structures result in the global minimum of the energy. So it is energetically favorable to this monomers come here and form the acid. So the molecular structure is comparatively the most stable and the most stable state. But there is possibility of exchange of its monomer between this non-acid and the acid state. But it occurs equally. So there is no rate change of its structure. So over the years, in fact, last 4 decades, it recorded on a good amount of work has been done on this static activity for the development of different materials, biomaterials for different purposes. Now, another type of assembly, that is the dynamic assembly. The structure and property that evolve with time and require constant supply of a certain structure as well. So what happens here? We have the figures. They reside at the global minimum of the energy. They do not take part in the same assembly. But in order to make them self-assembly, they are coupled to a chemical reaction network. That is a network of at least two opposite reactions. One is activation reaction. So in this activation state, the energy is supplied, the molecules get activated. So goes from here to here. Once they get activated, they can become acid. Here, they can become acid. Then what happens? Another reaction, that is basically opposite reaction, that is called the deactivation reaction. That actually deactivates these building blocks and the assembled structure, leading to the dissipation of energy, and the system comes back to its initial precursor state. So assembly breaks. So this is also called dissipative assembly because they dissipate energy during the reactivation process. Now these structures are these building blocks sustained as long as you have a supply of energy. Once you stop supplying energy, these building blocks collapses here corresponding to gas state. So assembly breaks. Now, so here the building blocks are intrinsically unstable. This is unstable, and they spontaneously reverse with origin state unless you are supplying energy. The dynamic state, means this state, is not the lowest energy state, so it's somewhat in between. And the structure and function sustain as long as we have the source of energy. Now, because of this field dependency, the structure is transient, highly adaptive, and shows time scale order. Now, biology actually adapts this type of cell assembly and to develop different types of interconnected structure for performing different biological activities. One of the typical examples is fire. So basically, distributed assembly is the hallmark of living system. Once equilibrium is reached, life is over. So this is the example of microtubules. So microtubules are an important part of the scientific system in maintaining cell structure, function, and other concepts. I'm not going to detail, but what happens here, the GTP, that is monocyte triphosphate, that acts as a fuel or energy source. When the binds this TBD protein, the TBD protein gets activated and they form the assembly. And the assembly sustained as long as they have the supply of the TBD. Once the supply of TBD is stopped, this structure does not get stabilized. So basically, the system consumes energy, forms assembly, does the function, and then they dissolves. So the kinetics of this activation and deactivation determine when the microtubules carry out the function. This is the image of the microtubule activity from the group. So we would like to develop a similar system or basically to mimic the features of the microtubules and the living assembly so that we can create synthetic type of material which shows lifelike properties, the features. We can capture the feature of light. So when we're going to create light, we are capturing the features, whatever features light has. Some of the features, if like capture using this type of synthetic acid. Now, what is the importance of this system in synthetic materials? So let us consider here you are adding. Once your fuel is added, your acidic forms. Once acidic forms, they can show you material property. So over the time, as the fuel gets consumed, your material property gets off. And again, if you are adding a fuel, the material property gets off. So repeatedly, after adding fuel, you can sustain your system. So basically, the assembly as well as the material property are the function of time. This energy dependency allows the system to be highly dynamic and the interesting properties like adaptability, auto-configurability, and stability. So another important thing is that when we form the assembly, where we form the assembly, and how long the assembly sustain can be controlled using this assembly or building assembly. Okay. So in our research, what we need the building blocks for creating such kind of assembly. We focus on short peptides. We synthesize our very short peptides, dipeptides, and we use those peptides for our as our building block for the creation of substances. So the peptides are typically the polymers and amino acids. So the amino acids joined together by an amino acid and so depending on the number of amino acids there, they can be called as dipeptide, and so on. Well, peptides, cell assemblies over the years have been studied of nucleic peptides. So peptides cell assemblies can structure they have apart. And I think now coming to our actual goal. So based on these, we have developed we have synthesized short. This is the content containing a value, E for value, L for leaves, and B for expert. And we have incorporated one pi system, pi technology system that is what design for the national. These pi actually serve as a plural code and it shows aggregation diffuse. Here there are two properties. Diffuse, that is one is aggregation and distribution, another is aggregation or building. So aggregation and distribution there are some flow code. When they aggregate, their emission property enhance the emission intensity enhance that type of molecule. One of the molecules, this one, that is enhanced. This shows aggregation intensity. But some type of molecules there, it shows that is aggregation cause quenching. Means when the aggregation forms, the emission intensity quenches. So here we are using this NIMOT that shows aggregation reduced heat. So we have this peptide and we have this we have taken our solution, we have maintained the pH of the system in such a way that each terminal remains as carboxylate, means diamagnetic charge. So because of this diamagnetic charge, the whole molecule is soluble aqueous medium, the medium that is pH6 in water. So the idea is that when we increase this, so it is a simple condensing agent that converts this dicarboxylate into a cyclic anhydride. So the idea is that when we use this EDC as a fuel, what happens is EDC converts this dicarboxylate into a cyclic anhydride. Once the cyclic anhydride is formed, the hydrophilicity of this molecule is lost. So once the hydrophilicity is lost, the molecule self-assembles in water, that is hydrophilic. So once it forms self-assembly, what happens? It can show changes in its photophysical properties. That is right. At the same time, this cyclic anhydride is unstable in the working medium, that is in aqueous buffer. So what happens is simultaneously it hydrolyzes and comes back with corresponding dicarboxylate. So thereby breaking the structure and coming back to its initial solution state. So this process state is called deactivation state and this is the activation. Now we have the activation, the rate of this activation is faster than this rate of deactivation, then we have the transient existence of this molecule and the acid. So when we add fuel, this rate becomes activation rate becomes faster. And when the fuel gets consumed, this rate becomes faster. So what happens when fuel is consumed? This comes back to here and the assembly breaks. Okay. So if you think about, so we tested our hypothesis. What we did, we have added. So initially as the t equal to 0, you can see a clear solution. When we add ETC, in 7 minutes you can see there is an appearance of some acid. And suddenly become carbon. Over the time, 60 minutes, because of this action from acid, then this undergoes hydrolysis and comes back to here. So acid breaks. So within 60 minutes, the acid breaks and solution becomes clear. Again, when you add this process can be repeated several times. So our hypothesis work. So then we tested this acid formation using the translating method to the absorption. So when we add fuel here, the translating suddenly increases. Then over the time, the translating gets decreases. Because this when we add this gas forms, that actually creates translating. And once the acid breaks, the translating goes down. So again, when we add fuel, this process can be repeated several times. So important thing is that this formation of the solution to assembly and again coming back to this solution, it is actually one cycle. Here. Okay. So this cycle or the lifetime of the assembly can be controlled by the concentration of the fluid or that is the EDC. So initially we take 0.5 number of EDC. You can see the line here around 0.2. So as we increase the fluid concentration, the lifetime of the assembly. So basically, how long the assembly sustain can be controlled by the EDC concentration. Then we check how the emission properties. So here, initially the molecule actually exists as sign emission. When we add this, it's turned into strong, and over the time, this slowly, as this analyte goes back to the cyclic state, this solution again comes to its original sign. So this is the initial state. So you see here, this is the initial state equal to 0, where the initial maxima is here, that is corresponds to this star. When we add this, the initial goes here, it will get to region, and slowly it goes up and it goes down. So that is also similar representation of similar of this. So here sign and again coming back to sign. So again, we can repeat this process by multiple time addition of EDC, multiple repeat. We add initial, the initial EDC goes here, then it goes down, again you add EDC, goes up and goes down. So, but you can see here, what is the validity measurements. Each cycle is not coming to the previous state because of this, because of the formation of the press. Whenever we are using EDC, EDC is getting consumed and one waste is produced, that is very EQ, and this that waste is hampering in this cycle. Again, the lifetime of the cycle can be controlled by EDC. So initially, when we have a low concentration of EDC, lifetime is low. As it is can go to high, lifetime is system. So here EDC acts as a programmable handle to switch between different time. Basically, you see, we are not at all touching the output. We are touching only, we are acting on the terminal part of the die, and that controlling the whole assembly cycle. So, when we discuss details, I show here. So, our idea, our hypothesis was charge utilization was the main reason for this condition. So, in order to what we did, we have added eight. So, this is our guide. Already one. When we add HCl and protonate is carboxyl carboxyl. Once the carboxyl is formed, the hydrolysis is reduced and the molecule acid product. Here solution, when we add it became acid. Same type of change can go with the emission spectrum, emission color. So this is the charge inflation responsible for this type of change. Again, the charge inflation we have shown using sodium chloride. Basically, sodium chloride acts. Sodium acts screening the charges. Then what happens when it screens the charges? It creates forms and or a complex which actually does not basically it does not allow the repulsion between the charges of when one molecule right outside is coming here or negative the charge, it does not allow. So yeah, so if you see here, it's a negative charge, another molecule, you try to come here, it repels. But if you utilize the charge with sodium chloride, the repulsion is stopped. Once the repulsion is stopped, the non-covalent. So basically the idea is that charge utilization is responsible for this type of change. Well, then we got can we use this transient color projection for some sort of application? Yes, what we did, we we have taken one agarose gel matrix and we have incorporated agarose our peptide molecule into this agarose gel model, gel matrix. Then what happened? The whole gel shows the cyan color. Now, when we then what we did using a plain product, we have applied a here. Whatever we have applied here, they are cyclic analytic forms. Thereby, it's on the assembly and the color change. So, especially you have applied, and over the time, you can see from basic treated, and over the time, the basic get automatically erased. So basically, it's a spatial control. So in the space, wherever you have taken your changes occur only at that point. Again, you can draw. We have drawn here one round parts, which again over the time self-erase. The same plane which has drawn one leaf or spherical and spherical again is for the time. Importantly, that whatever changes occur in policy can be visible under the UV lamp. So if we visualize in naked eye, we cannot see it. But we see under the UV lamp, we can see it. Well, this is what has been published here in Excel body. And based on this work, I have been featured as an emerging investigator where they highlight the exceptional work by an artificial. So this what we have previous work. What does I talked about? This is chain between only two colors. Now, what can be created on demand multi-color division which is distributed on the. So this is quite relevant to a natural phenomena that is fire difference. But the emission of light by giving on these chemical reaction. So if there is light is used for different functions, it will complete current and defense mechanism. So only measured in most of the cases, this type of ions is found in various. And many of the cases what happened, they show only one color. In some cases, they be more than one color that can be one another. So in order to create this type of on-demand multicolor emission, what we do is a simple tool that is called cluster resonance energy transfer. So what happened? What is cluster resonance energy transfer? This is basically a non-linear energy transfer that occurs by dipole-dipole coupling from excited state donor and the ground state events. So basically, when you have one donor and you have one acceptor, if they have good spectral overlap, that means if we have the spectrum of the donor emission overlaps with the absorption spectrum or excitation spectrum of acceptor, then get on. So usually there is no good overlap, but here you can see good overlap. So there is possibility of energy company instead. So whatever energy donor emits, that is being accepted by the acceptor, and acceptor actually emits. Another character is that which is controlled that is the distance, donor acceptor distance. So when the donor acceptors are far away, when donor acceptor are very close within the diameter, then thread off. Same thing in the case of multi-state. So that is in the case of multi-state, yes, this is donor and they are acceptor having spectral overlap and they have closer proximity. So when you are to excite this donor, that excitation energy is transferred to first donor, then second donor, then third donor, then this is donor. When you excite this donor, this energy transfer to first acceptor, second acceptor, third acceptor, and the third acceptor actually emits. So basically, we excite the molecule with the donor excitation energy, but we got the emission from the final acceptor. Then we got. So if we can create a temporal thread, we can create some sort of multicolor. So idea is that if we take one donor molecule and one acceptor molecule, having good spectral color. Now, using the chemical equivalent approach, we can measure, we can actually control the distance between the donor and acceptor dynamically, then we can create dynamic field. So here is that. So we have taken one assembly unit containing the donor and another acceptor. Okay. So when there is a solution, donor and acceptor they are forever. So there is no credit. But when we add our critical field calls the assembly. That assembly actually brings donor and acceptor to certain proximity. Then then the thread occurs and the acceptor gives emission. Same thing. And at the simultaneously what happened? As the fluid gets consumed, the donor acceptor distance goes away, goes increases because of this assembly break. So as the assembly breaks, donor acceptor goes away. Then what happens? The thread efficiency decreases, and after some time the thread becomes off, and it comes back to each other. Same thing happens here in the case of the sequential state. So we have one donor and two acceptor. We actually what happens here? Donor gives energy that is transmitted transfer to the first acceptor, then to the second acceptor, and the second acceptor actually release the emission. And simultaneously, what happens? When the field is consumed, the acceptor breaks and comes back to this state, and the rate down. At this point, we thought that instead of taking normal assembly, we take hydrogen, which is a very higher degree of assembly. Then hydrogen can bring donor acceptor is very close proximity by their compact environment. So the hydrogen is basically an extreme case of higher assembly. Then what we did was design another peptide having the similar phase. So when we add the assembly, this and the anhydride assemblies form hydrogen. You can see the always nice fibers, network fibers. And when you add VDC, you forms gel and over the time the gel breaks down. So you can see here is the video. You can see the video here. We have just added field. Initially it was clear, just after adding the field from the assembly and again become gel. And over the time you can see the translucent gel become clear cell. You can see here. Just after a few seconds. Okay. So then we calculate how much the kinetics of this reactor. What happened? So usually you have this precursor of a you can see the conversation, the graphical is it and over that it comes back to digital state. So this is going the consumption of this one over the time again it is coming back. The next thing is the simplification how this activity is changing this color. So this is the here. This color is going from green to blue and the change from back is the same. And one of the meters. Let's see. Yeah, this video will add some start becoming turn blue. Then so yeah, you see the speaker. Yeah, usually the the speaker was here, the speaker was here. That's the condition of the material. The intelligence goes up and again goes up over time is of that. It's a state. Then in order to make political, what we have done, we have taken accident one. What is this? The accident one. Another accident. There is one. Enough. Some of this will be returned. This will be here. So first we check whether we are showing this spectrum over. Yes, we have checked. We have the spectrum over. So this lunar and this accident. Lunar emission has the spectrum over and accident. So this. Similarly, these and these two spectrum overlap, and yeah, these and these they have four spectrum overlap. So the condition of spectral overlap is satisfied. Now we have to satisfy the condition of distance between the neuron axis. So this slide become a bit complicated, but I will try to make it easy. So initially what we have this molecule, this one showing the green emission, on the inside active system and the parameter, it shows green emission, and when we add this transformed into this, and it shows blue emission, the emission parameter for 10. Now then what we need here, in this case, we have donor 1 and donor 2, accepted 1 and accepted 2. What happens? In the solution state, despite having this spectral overlap, they will not show any trend. But only at this, what happens? There is a transport energy occurs because in the when it actually forms, these accepted molecules comes closer to that of this boron molecule, and the energy transfer takes place from blue to this BBP, then BBP to the nitrogen, that is the and it shows the emission of emission 30 nanometers. It's a radiator. So, and so over the time, what happens when these actually breaks, the thread interaction also stops and it comes back to its original reconnection. Now, if we see this one, so first we tested stable line. So, initially, we have taken our peptide and we have taken a liquid. So, there is no this color is green. Only added is just on gel and the color. So red occurs and the color transforms into a white black liquid. And number the type of color changes to light yellow, UV green, and as the assembly breaks for the time, it's not black. This video could be interesting if it works. You can just turn into white. Then it is breaking. So this is the substrator. So initially it was here. Then just the tradition, the segment looks like this one, this one. So this P is coming from the red, or that is mid of this one, and this part is coming from this. So when they mix together, they are giving a white tridimension. And over the time, as the ratio of this one and this one changes, the resultant color is also changing. And finally, consistent breaks come back in constant in color values. So then we can say it's spatial, right? That is donor to activator, activator to activator. So here it makes all of it. There is no only the color coming from this one. So we have it. They forms donor activator comes over. What happens there? They offer and the offers is ready design. So over the time, this generates and the mixing of color comes back is okay. So then again we can say the directed means here to here, though they have no fatal still they can look pretty bad. So initially the green only and this become purple and over the time color changes to they come back to green. So again this video it works. So personally statement is there. Initially, the statement this the statement where it is on the field, just the direction of the field, this is coming to donor and this part is coming to the thread. So, and now they are mixed together, they are creating different colors. Now you can see the third one that is directed. You can see the video. So we decided the color changes to do it in purple. This is the part. Statement is the this part is the red. Well, so can you repeat the system using multiple activities? Yes, you can. So this is we can confirm that so here we add this is program, change program, and programs again. This is repeated several times, and this activity is applicable for all the systems. Other thing is that we also determine the region of this condition of the color. When whatever results required, that yes, we are reached 200 kilograms of PC. So that is we have around 1 hour of time of the system. Now here we will add there is around 50 milligrams. So what happens? The cycle completes before 20 minutes. So as the idea is greater the constant here, greater the right time. Okay, so then then we have so we actually the cascade of events in a bed. What happens? You are adding so you have the initial molecule When you are adding to this, it transforms in this one. When it forms, it adds acid and it forms gel. So when it forms gel, the color also changes here to here, and so when the gel forms, it can interact the accepted molecules to the. So basically, gel acts as a distance monitor between the donor and acceptor as well as the emission monitor. Then what happens? Once the donor acceptor comes closer, trade emission occurs, and because of trade emission, that is also time dependent, and then because dependent on the distance between the donor and acceptor, that is controlled by the fuel. So the continuously captured emission and the distance result in an oscillating energy transfer process, which ultimately produces in multicolor luminescence that is controlled with timing. So this is the whole sequence of process which in multicolor interpreted can simply be initiated and programmed to be touched by the fuel in a finite time scale, then it is optimally on mark. Finally, a system strategized with what emission current velocity, when we contact how long we want, can be achieved by manipulating the additional fuel and the different combinations of fuels. Then we have created again the synthesized system. We can write on it which we can get the best product. So you can see that in this case, we have created a information encryption system where the right information can be obtained only at the right time. Otherwise, we will lead to either fake information or no information. So what we have created one matrix, and we have filled this well with different combination of donor and X. But when we add the field, actually the combination of the donor and X will create some. We can remove those colors and bring their appearance. So let's say after 2 minutes, this one go, and let's say it's take the conversion. After 40 minutes, let's say other set of colors appearance, let's say writing conversion from right. And again other colors forming, and now the lines from black is black color. Now, if you want to get the right information, you have to whatever color combination you say right, you have to read this combination at a particular time. Otherwise, let's say if it is a right information, you have to read the color combination to get 40 minutes. If you read the active minutes or 45 minutes, you will limit the information false. If you read at 60 minutes, you will not get it. And you will not get anything. So, yes, sir. Yes. So basically, we have developed a diagram. This properties function that convert with time. And the paper, the high impact journal, some local news take the diagram to work. Well, so what are the challenges of basic physical diagnosis? First challenge is the system design. You need to develop a system that responds to your activation and deactivation times. Second thing is that we need the orthogonal process activation and deactivation. Activation rate is faster than the deactivation. Three possibilities are there. One is that you are adding a here. You are getting activated, but it does not accelerate. So in that case, what is happening? If your repulsive forces between molecules is higher than the activity force, then what happens? Molecule does not accelerate. Despite we have activation and deactivation time. Another possibility is there, particularly attractive force is higher than the repulsive force, the molecule itself accelerates and not undergo it. So it's like standard. Second, that is this one, what we basically looking for or want is that responsibility and attractive force, they are somewhat similar. Slight imbalancing with that symbolic and attractive forces can be activate, can be assembly or different. Third thing is that waste ingredients and combination. So each step, whenever you get the fuel, that fuel is producing some sort of waste, and that waste actually in the I did not that some people are using some software that will be light or some gaseous waste from a produce. Like if some people use we can read where there is ammonia or person. That is what happens. The worst thing of players can be attacked. And according to the record time and this, you see, this is a diagram and changes from figures. So you need more like usability techniques for the analysis of the picture. And the last one is that so far this field and somebody new and so far the system has been developed will be focus on the event system development. Real time application still lacking. Uh, some other actually work that is related to static network for this. So we have developed this static, we have this peptide, it's from hydrogen. Just simply this hydrogen going to apply force, the hydrogen melts, and if you remove the force, the field, it's on the tail. And it's highly stable or different. So what we did, we have used this hydrogen for removal of toxic dye of this one. So this is a dye, or a dye toxic dye. What we have done, we have taken our gel. Over the gel, we have the top to gel effect. The gel acts on the dye. Here is the gel. You can see here. So this is the dye. The gel. On the dye is red color here. And it's the hydrogen. After that, is the dye at top. Now we have developed this spectroscopy. We have taken the formats and this photon is put into the our gel. So what happens is we dip this photon into a dye element, the automatically absorb the dye. Here. You can see here the rotation the gel coated photon we have taken and it is empty. Now we take the normal result gel with the dye. Again, what we need, we have prepared a filter. What is the filter or gel? So this is a solution, this is a non-filter, all the solution charge passes to the forms. But when we have taken this gel filter, these are the beams. And the radio works the dichotomy solution and the gel filter paper. This is not published in the Kumar inside Conversely. This is developed. And some other peptides are developed and those. So when we discuss these are muscular and muscular. So finally, coming to the most important side, that is the acknowledgement. So like acknowledgement speaks and the second part there was the this therapy work. These therapy actually from here and I got the group member. So because we have icon over value sources, all the rules and experiment conducted from the lab and here. And experience. And my collaborators, we are following different and we can photomaker and we developed provided is laboratory is used for the experiments. With the and we are getting the my current system and binding and it for is the program. Yes, this colors. There exists literature is for the basic reaction. In short, is our basic reaction. Is the basic for molecular or automolecular reaction. That is the initial first one. I think that so that was nice. So when we started from the that time it was important. So there is a solution and it's starting so basic. So all these three government statements the really not experience. That is coming from the education provided. Or when the research for the then we are what the time. Perfect example or extraordinary ingredients is there where the acidity and functions are excited from control data. Whenever you need only that different blocks there, we enzyme, so we try to create it. That will be detailed. Some of the features in our and store the kinetics. It's in the periodic system. Actually, it's after a differentiation. Now, you need to differentiate, not you cannot decide the periodic. Have you ever tried to model this by two couples of nonlinear master differentiation? You know, the densities. It's going to add one extra term. That is the addition of fluid. Otherwise, there is a kind of dynamics fluid. Yes. And we have such a activation. So I have said delivery English and okay. So if any of the line is done. But in this presentation, though it is not involved in illustration, but I think the color is that. The second is one time is this crypto chemical. So there are two possibilities. Either you will or you want to if it is the second one, I am at this out. Okay, fine. Let us help this very